Okay, I was getting confused.
Okay, guys, so we're going to go ahead and get started. Hold up, Oscar, let me set you up. So we're going to go ahead and get started, and what we're doing today is we're finishing up our DNA model projects. Remember, the objective for this project was demonstrating to me how DNA structure rules work, how DNA base pairing works, and we were given an unknown identity. And our job today, Oscar, is we're going to try to guess to see which person you are by reading your code. Okay, so what we need from you, Oscar, is we need you to walk us through your DNA code, and then all of us, we're going to look at the DNA names that I gave you guys, and we're going to try to guess who you are. Okay, so when you're ready, Oscar, please share your screen with us. Okay.
I have it on the paper. I wrote it down, the codes. Or do you want to read it off the model?
Why don't you write it on a piece of paper and just hold the piece of paper up for us, because it looks like
Oh yeah,
kind of
I already have it. Yeah.
Okay. Can we look at that, guys? So, can you read it out loud to us, Oscar, from top down?
All right. So it's thionine.
Or you can just say the letters. You can just say TGC or something.
Okay. So it's T G G T C T T A G T T G T A. Shut up.
Okay, so continue sharing your screen with us, please. Oscar, continue sharing the screen with us.
Wait. I am.
So just leave it there for a little bit.
What's happening?
Okay,
Since you guys can't really see like the letters, like that's the color code. The red is T, the green is A, the purple is C, and then the yellow is G.
Okay.
Remember you said Thanos isn't a real name? Oh shit, wait,
Okay, so who would like to take a guess? Who do we think Oscar is?
I think it's Alexandra.
What do you say that is?
Well, the sequence that he gave us, I broke it down and I found what would it be, the trait, the traits, yeah, for each one, and then all 5 traits match to those of Alexandra.
Perfect. Thank you. Is she right, Oscar?
Yeah.
Absolutely. Awesome. Thank you, guys. All right, cool. So, Oscar, like everyone else, like I told you, I need 3 pictures from you. Okay. I want a picture of your model. I want a picture of that color code that you showed me, and then I want a picture of the code reading all the way down. Does that sound good, Oscar?
Okay.
So when we end today, Oscar, please send me those 3 pictures as an assignment to the model. To the assignment called DNA model. Okay.
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 All right.
All right. So that's Oscar. Who else needs to present today?
I do.
Okay. Okay. So go ahead and share your screen with us, please.
What are you doing? Just curious, why do you have your AirPods
Go
on
ahead
like
and
that?
share your screen, please.
Why do you always have them like that?
Who's presenting next? I need you to share your screen, please.
Do you want a headband?
Can I go?
Yeah, go ahead, please.
Okay. Okay, that's it right there. Do I just show like the sequence?
Yeah, can we first see your sequence, April, and then we're gonna guess who you are, and then I'll look at your model, okay?
Okay, that's the sequence, the top one, and the bottom ones are just the ones that match to it.
Okay, leave that there for a little bit so that we can figure out your code. So just leave that up for a little bit. Okay, who would like to take a guess? Guys, who would like to take a guess? Who is April?
Let me see.
What, you're gonna play Fortnite? You're
Is
gonna
it
play Fortnite?
Eduardo?
You're gonna play
Eduardo.
Fortnite?
Is she right, April?
Yes.
Okay, good, good. Awesome. Awesome job. So again, April, make sure you send me. Now let's go ahead and look at your model, please.
Okay.
And explain the color code that you did, April.
So the T is orange, and that matches with A, which is purple. And then the C is white, well, sort of a white, and then it matches with G, which is green.
Perfect. And go all the way down for us to see. Nice. Awesome. And so did you actually, April and Oscar, my apologies, Oscar, did you have any trouble getting the model to stand up or like to stay in shape like that?
Yeah, it was
Standing
a little
up,
hard
yeah.
to get it to stand up.
How did you overcome that challenge, Oscar and April?
I stuck it to like a foam ball, and that seemed to work.
Okay. How about you, Oscar?
So like I use tape for like the sides to like hold in the popsicles.
Mm-hmm.
And so I got like some metal wiring, and I just surrounded it to hold it up. But like for the bottom to keep it up, I just got some toothpicks and then just hope for luck that I didn't fall.
Awesome. Awesome. Oh, it worked. Okay, cool. Cool. Anyone else want to present that didn't get a chance to present on Monday?
I didn't present.
Okay, ready, Thanos? Yeah, is your camera working?
Yeah.
Okay, go for it.
Anyway, hold on.
I see blackness.
I know. Fine. One sec.
It's okay, I didn't pay my Wi-Fi bill either.
Are you ready to share your screen with us, Thanos? You have to hit share screen so that we can see what you're showing us. Thanos, are you there?
Yeah.
Okay. Well, I see Peter Griffin, but I don't see you.
Stuck.
Oh,
It has a green screen. I don't know why.
Yeah, all I see is Peter Griffin.
Yeah.
I see you now. Why don't you take that? Why don't you instead of doing the virtual background, Thanos, just take off virtual background and then just share your camera screen with us.
I don't have any. Oh, okay.
There you go. Okay, and then flip it towards your model.
All right.
There we go. Perfect.
Please, man. Can you guys see it?
Oh, you just gave us the name.
Yeah, oh.
It's okay. But let me see your DNA code, please, and explain to us why you're Kenneth. How about that, Thomas?
Okay. I'm Kenneth because AGG is the coding for blonde eyes.
Hold on,
No,
Max.
wait, not blonde eyes, blue eyes. And then
Max, why are you
GCT.
sharing your screen? Hold on,
Oh
Thanos.
my bad. I was like messing around with it.
No, please don't share your screen because Thanos is presenting. Okay. Go ahead,
Okay.
Thanos. Sorry, go ahead, Thanos.
And then GCT is the code for blonde hair. And then TAA is the code for dominant right hand. And then GTT is the code for height. I mean, for being tall. And then CAT is for having pointy nose shape.
Okay, perfect. Thank you, Thanos. And even though you gave us the answer already, it's okay because you have the sequence and you told us why you have the particular trait. So do me a favor also, Thanos. I want a picture of the model. So you're going to show me the model now. Then I want a picture of your sequence code. And then I'm going to match that up to Kenneth to see if you're right. Okay, so please, can you show us your model real quick? Thanos, can we see your model again, please, all the way down?
Right?
There it is. Okay, cool. So looks like, what materials did you use, Thanos?
I used Q-tips and tape because I didn't have anything at all.
Okay, and what did you, so supplies obviously are your biggest challenge, but any challenges
Yeah.
getting it to stay twisted or getting it to stand up?
Getting it to stand up wasn't that hard because I had a platform, so I could like just use tape on it.
All right, cool.
And then, yeah.
All right, thank you, Thanos. So please, again, Thanos and Oscar and April, send me 3 pictures, right? Send me the model itself, send me your DNA codes, like all the letters for the person that you picked, and then your color coding system. Cool?
Okay.
Okay, thank you guys. All right, so we're gonna go ahead and move on now. I'm gonna share my screen with you now. So let's go back to Schoology. And can you guys see my screen? Can you guys see my screen?
Yes.
Okay, so what I'm gonna walk you through today is, remember I told you on Monday that I restructured the class, and what we're gonna be doing for the rest of the year now is all of the stuff we cover are gonna be found as modules, okay? So we're wrapping up the DNA module. For DNA module, guys, here are the notes that we're using for this. Here are the assignments that we already completed. We already did that reading assignment on COVID-19. We did the reading assignment on DNA structure. You guys already did your DNA quiz. Remember last week, or actually it was almost 2 weeks ago, you already did that. You already, and again, we're finishing this up. I'm just reviewing with you. We have one discussion board we did, but then you have another discussion board that you're going to turn in this Friday. Okay, so what I want to talk about now is DNA mutations, and what I'm going to talk about today, you're going to use to answer the discussion board, which is due on Friday. Okay, so the other part of the module is the summative assessment, and that's what you guys all just presented to me. That was your DNA model, that little building thing you did. Okay, so thank you all of you that submitted your projects on time. And again, if you weren't here yesterday, I know Cynthia wasn't here, please just submit it to me or let me know when you want to present it or how you want to present it. And that's what that is, okay? So let's go ahead and look at the last part we need for this module, which is the mutation note. So if you could all go ahead and click on DNA notes, and there is a section we need to review. Well, there's actually 2 sections we're going to review for chapter 8, and that is going to be 8.3 and 8.7. 8.7 is going to be used for the discussion board. 8.3, I'm going to actually introduce this to you in a Zoom breakout right now. Okay, so what we're going to do today is I'm going to put you into your Zoom breakout groups, and you're going to watch a video on DNA replication. So in your in your website folder, there is a video I posted for you called DNA replication. So same as we do, I'm going to put you in Zoom groups, and if one person could share their screen and watch the video, after you've had a chance to discuss the video, we're going to come back as a class and talk about what DNA replication is, and then I'll give you some brief notes on it. Sound like a plan, guys?
Mm-hmm.
Okay, so here we go. So again, the video is called DNA replication. It's found in your pink website folder. So here we go. I'm going to put you in groups now. You're going to get an invite from me. So we'll just do 4 groups, okay? We'll do 4 groups of about 5 people, and just one person, please play the video for the group, all right? Here we go. You're going to get an invite now.
Yeah.
All right, bye guys. I'll see you in your groups.
Oh, it's true. I accidentally left.
Oh, it's okay. I'll put you in a group.
Okay.
Did you get the invite?
Oh yeah.
Okay, I'll see you there. So who's playing the video for us?
I am.
Okay.
I have to get to it first, though. What was it called again?
It's called DNA replication. It's the very last video, Don.
Yeah, I see it. The PBS one?
Yeah.
All right, let me share my screen.
Yeah, that's the one.
All right. I don't think audio works.
Oh, what the heck? Can you guys hear it?
No.
No.
Are there subtitles?
Yeah, there is. But
Well, I could hear, but it like glitches out, like barely.
yeah, I can't hear it at all.
I can't hear
Even
it.
though I'm not sharing it.
how about this? Pause it, Dominic. Can you guys see if you can see it individually, though?
Okay.
Try that.
All right.
See if you can play it individually, guys. And if not, then I'll just play it for the class. I'm sorry. We're still learning how to do online stuff, right?
Yeah.
Let me know if you guys can hear it individually, okay?
Which one is it?
One called DNA replication. It says PBS DNA replication.
Oh yeah, I could hear it.
Okay.
I could see it individually.
Okay, my bad, guys. So can you watch it individually, and then I'm going to check in with you guys, okay? I'm going to go check in on the other groups, and I'm going to let them know the same thing. Sorry about that. Hey, guys, are you watching the video? Hello? Guys? Are you guys watching the video?
Yeah.
Okay, thank you.
One counterpart, one base.
Can you guys hear the video?
Nope,
Kind
we
of.
can't.
Oh, you know what? I think the problem is, guys, we just discovered this in the other groups. For some reason, when you play it as a group like this, it kind of glitches out. But if you play it individually on your own screens, guys, you can hear it. So can Noemi, can you actually just exit out of your share screen mode? And if you guys all watch it individually, that way you can hear it. Okay, I'm sorry, guys. I think I put the link up incorrectly. So please watch it individually, and then I'll come back to this group. Okay. Sorry, guys.
All right.
Sorry. Hey, Victor, you don't have a group?
No, it kicked me out.
Oh, it did? Okay, sorry. Which group were you in?
I forgot.
Okay, I'm going to put you in one right now, okay?
You're right.
You should get an invite now.
Hey, mister.
Hey, guys, are you watching the video?
We tried to screen share, but none of us were able to do it properly. When I did it,
Yeah,
the
yeah.
audio
You know
didn't
what? I'm so sorry, guys. We just learned this in the other groups. If you share screen it, it doesn't play. You have to watch it individually.
Oh.
Yeah, I know. My bad. I think I uploaded it wrong. I'm sorry. I'm still learning how to do all this online stuff too. So can you guys all watch it individually, and then we'll talk about it as a class, okay?
All right.
Okay.
Okay. Thank you, guys.
Sure.
Oh,
Are you guys watching the video still?
Yeah.
Okay. Are you guys watching the video okay?
Mm-hmm.
About how many more minutes would you say?
2 more minutes.
Okay. I'll break us up in about 3 minutes.
Maddening face only finding counterpart. Likewise, one side of these bonded nitrogenous bases are called base pair. GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you. So AGGTCC means something completely different as a base sequence than, say, TTCAGGT. Human chromosome 1, the largest of all of our chromosomes, contains a single molecule of DNA with 247 million base pairs. If you printed all of the letters of chromosome 1 into a book, it would be about 200,000 pages long. And each of your somatic cells has 46 DNA molecules tightly packed into its nucleus. That's 1 for each of your chromosomes. Put all 46 molecules together, and we're talking about roughly 6 billion base pairs in every cell. This is the longest book that I've ever read. It's about 1,000 pages long. If we were to fill it with our DNA sequence, we'd need about 10,000 of them to fit our entire genome. Pop quiz. Let's test your skills using a very short strand of DNA. I'll give you one base sequence. You give me the base sequence that appears on the other strand. Okay, here goes. We got a 5' AGG.
3' TCCA.
Time's up. The answer is 3' TCC AGGC 5'. The other words is not too complicated. Since each nitrogenous base only has 1 counterpart, one base sequence. But it's magic sequence. So, could I make the same base strand of that other this one to sugar phosphate backbone, nucleotide bases attached to it. But there are 3 major differences. One,
Have you guys gotten to the section on DNA replication at the end?
I'm finishing up with the video.
Okay. The most important part is the last part, guys, the DNA replication. The intro is what you've already talked about and you did on your model. I want you to watch the last part, please.
I was wondering, who figured all this stuff out? How big was the brain? Well, after all of that was this snotty gray stuff.
Okay, so we'll break up all these grooves. All right, so you should all be coming back here to class. I apologize for the video not playing properly. I think I linked it wrong. So we'll just talk about it as a group. And if you didn't get a chance to finish talking about it, it's okay. We're going to talk about it now. So everyone should be leaving their Zoom breakout groups. And if you didn't finish the video, that's okay. I apologize for the way that I linked it to you guys. It didn't allow you to hear the audio when you watched it as a share screen. You had to watch it individually. So sorry about that. But we'll go ahead and, we'll go ahead and get started. So for those of you that did finish the whole video, the part I really wanted you to focus on was decontamination, which was the last part of the video. The beginning part of the video was
The car.
this.
I mean the tires.
Mia, did you say something?
No, I'm sorry.
Okay. So what I want to talk about is the last part of the video, the DNA replication part, not the beginning part of it. So for those of you that did watch the whole video, can you tell me, like in your own words, what is DNA replication? From the video, anyone?
It's when you keep on making copies of the same DNA strand.
Mm-hmm. And why do we need to do that?
To
Think about it. Why do we need to do that? So far, we've only been talking about what DNA is and what base pair, structures are and how you built your model. But now the question, Felipe, and this is for everyone, not just Felipe, why do we need to do this, do you think?
To probably like repair damaged ones.
Exactly. Thank you, Oscar. So when we repair things, when we need to make more cells, we need to also make DNA. So I'm going to share my screen with you now, guys, and follow along, please. And what I want to talk about is what that video was about. And again, if you didn't get a chance to watch the whole video, I'm going to share with you right now what that video was about. So I'm going to briefly talk about section 8.3. It was covered in the video, but I just want to highlight a couple of the really important things. So first of all, you don't need to copy this. Let me just explain this to you. So in the book, DNA replication is also known as the semi-conservative model of replication. And what that means, and again, this is what Watson and Crick, which we already talked about, they're the guys that presented what we refer to now as the semi-conservative model. What that means is there is a parental DNA strand, and the parental, if you look at my model here, a parental DNA strand, think of it like as the original DNA, like Oscar said that we're going to, well, Oscar and Felipe said, we're going to use to make more DNA if it needs to be repaired, for example. So if this is a parental DNA, what's going to happen is this parental DNA will unwind or unzip, okay? And one half of this blue parental will be used here as a template. Do you guys know what template means?
Like a format?
Like a format, right? So we're going to use a part of this blue parental as a format or like something we can copy. And you notice there's one up here and there's another one down here. So we're going to use a parental or a template on the top and on this bottom to make a brand new DNA strand that you see color-coded in red here, okay? So for example, okay, let's say my parental DNA that I'm going to unwind on the top blue one, let's say it reads ATA. If the top DNA reads, I'm going to just type it for you, if it reads ATA, according to complementary base pair rules, what would this little red matching one be then?
TAT.
It'd be TAT, right? And I'm trying to type it here for you. It would be, say it again.
TAT.
It'd be TAT, and I'm going to color code it in red. Okay, so all we're doing is we're unzipping a parental DNA, and if the DNA template reads ATA, we're just matching up according to our base pair rules to make the other half, which in this case would be TAT. Does that make sense so far, guys?
Yes.
Okay, very good, thank you. So the purpose of it, meaning why do we do it, like Oscar told us, is because sometimes we need to repair stuff, right? So if you recall, do you guys remember the cell cycle? I know we did this back last semester when we were on cells, but do you guys remember the cell cycle? In the cell cycle, remember there were these phases that we called G1, S phase, G2, and mitosis. So mitosis we talked about a lot already, meiosis we talked about, but now I want to talk about this little segment we never really talked about called synthesis. In the synthesis or S phase, this is where all that stuff takes place. This is where we're going to unzip DNA to make extra copies of it using base pair rules. Okay, so this is where we're talking about. Okay, so this takes place inside of the S phase of the entire cell cycle, and this is where we're going to make more cells. Don't copy this. This is just a really, really complicated overview of what DNA replication is. So this basically represents this, just way more detailed, okay? So all that's happening is a parental DNA gets unzipped. We get 2 templates to make 2 brand new DNAs using base pair rules. That's what this is, just a little more detailed, okay? So in DNA replication, though, it doesn't happen all at once. It happens in steps. So these are the 3 steps of DNA replication. Can you read us, please? How about Melissa? What are the 3 steps of DNA replication? Melissa, are you there?
Yeah. Origins of replication and strand separation, priming and elongation, proofreading, and termination.
Very good. So I'm not going to go over all of the steps in detail here because you can just read this on your own. I just want to highlight the important stuff. So we'll start with just step 1 for today. And step 1, if you can copy this down, guys, step 1, like Melissa said, is what we call the origin. Do you guys know what origin means?
Like where it began.
Where it begins, exactly. So like Jaime said, origin means beginning. So this is just the beginning of DNA replication. So in other words, this is how it begins, all right? And it is also where the strands separate. So remember when I showed you that picture of it unzipping? That's all that we're going to talk about today. We're going to look at step 1. Please write it down. Step 1 is called origin of replication and strand separation. So give me thumbs up once you've copied all 3 steps so I can show you just step 1 for today. Just step 1 for today. Give me thumbs up once you've copied just the 3 steps here. Okay, thank you, Alexa. Thank you, Oscar. Thank you, Mia. Thank you, Vanessa. Thank you, Christian. Okay. Thank you, Maria. And again, all this is on Schoology, so you can review the steps on your own. But for now, we're only going to talk about step 1, like Melissa read us, okay? So step 1, here we go. And again, I'm recording this, so you can always go back and look at this. So here's step 1. Now, before you freak out about all the words, let me just explain. It's exactly what we just looked at in the previous drawing. What's happening here is the parental strand is unzipping. The parental strand is unzipping. So the unzipping has a name. It's called a replication fork. Do you guys know what fork means? Like when they say there's a fork in the road, do you know what fork means?
A split.
It means a split. It's a split, right? So step 1, like Melissa told us, all that's happening here is there is a split that takes place right here. That split, we call the replication fork because that's what fork means. It means split. And when that happens, okay, when it opens up, now we, like, let's say this that I'm pointing at here, let's say this reads ATA like in the previous slide. Now that we have unzipped it, if we use base pair rules, we can just match up more bases and make a whole new set of DNA from that unzipped template. Okay, so this is called step 1. It's called the replication fork. So this is step 1A, where we have a DNA being unzipped, and the name of that little Y shape unzip is called the replication fork. You with me so far, people?
Yes.
Now, check it out. Do you guys remember what 5s and 3s mean? I put a 5 over here, then I put a 3 over here. Do you remember what the 5s and 3s are?
Carbon numbers.
The carbon numbers, but what do they tell us? What do 5s and 3s tell us, Alexa?
They tell us where the DNA starts and ends.
Thank you, Jessica. It tells us where's the start and where's the beginning. Okay, please keep that in mind because it's going to be very important for what I'm going to say next. Okay, so that's step 1A, the replication fork. Now, you don't need to copy this. This is just a picture, okay? So the replications happen all over the DNA, and this is an actual microscope image of DNA replicating. So check it out. It doesn't just replicate the entire DNA. Like we saw in that video, DNA is billions of base pairs long. So it doesn't, if I just want to repair like a broken bone, like Oscar said, I don't want to repair and make all my DNA at once, right? We're not making a whole new Trujillo. We're only making the DNA instructions for that trait. So let's say, for example, the trait is found just right here, right? It's not the entire DNA. We just want to make that little piece that codes for that trait. So what happens, you can see the little pink arrow. It unzips and it makes a replication fork here. And all this opening up, this is it making more DNA. So it's only replicating what's found in this bubble. It's not replicating this stuff. It's replicating this. It's replicating that. So basically, it happens in small little chunks we call replication bubbles, not all at once. Does that make sense, guys?
Yeah.
I'll say it again. When we're making our DNA, I'll go back to this, okay? When I'm making a DNA in the S phase, remember, this represents a cell cycle. If I want to make a new bone cell because I broke a bone, I don't want to make my entire DNA. I only want to make the segment of the DNA that codes for that trait. And the reason you did your models where you coded for traits like eye color and nose shape or something is because DNA codes are just small little segments. So rather than making the entire DNA, we're only making them in small little bursts we call replication bubbles. So think of them as just the small little chunks. Does that make sense? It's the small chunks for only what needs to be coded. Does that make sense, guys?
Mm-hmm.
I'll put it in code, in bold. Only codes for segments needed to do repair. Meaning we're not making all of the DNA at once. We're only making, oops, hold on. We're only making the segment that needs to be repaired. And it happens in little segments called bubbles. And again, I'm recording this so you can rewatch it. Now, this is where I'm going to end. What was the name of that split again? What was the name of the little opening?
Fork.
The fork, right? It's called the fork. The name of the enzyme that causes it to fork or split is called DNA helicase. DNA helicase, which you can see in this picture right here, it's this little blue triangle. This little blue triangle, what it does is it causes the DNA to open up. And remember, in your models, you remember how you connected your nitrogen bases? Some of you use toothpicks, some of you use popsicle sticks, some of you use straws. What were the little sticks connecting the bases together? What were those?
Hydrogen bonds.
Hydrogen bond.
Exactly, guys, you got it. It's the hydrogen bond. So what DNA helicase does, in order for us to make the split, what do we have to break up?
The hydrogen bonds.
The hydrogen bonds, guys, you got it. So in step one, like Melissa told us, all we're doing is we're making the split. We're only making it for the bubbles, and we're using the enzyme called helicase to make that split. All right. Now, as this happening, you'll notice these little purple balls here. What these do is they attach and keep the strands separated and untwisted. So meaning these guys are just assisting in the process of unzipping. That's it. But the main enzyme at work here is called helicase. I'll pause there for you guys to copy. And again, this is being recorded, so you can always go back and look at it again. Okay, so I'm going to go ahead and pause there. The next enzyme is called topoisomerase. This one I'm not too concerned about. As you can see, when the thing unzips, you have like another enzyme that is kind of supporting the whole system, the whole DNA, I should say, so that it's not like unzipping and going all crazy. So this one, topoisomerase, is not as important. The one I really, really want you to know is called helicase, okay? Because that's the main one. So I'm going to pause there.
Yeah.
I'm going to pause there.
With Alina.
What I want you guys to do, guys, in the next couple of days, because we're going to meet up on Friday. Friday, we're going to finish the last part of this module, which is mutations. So in your notes, guys, everyone do me a favor. You have a section called 8.7 mutations. Between now and Friday, what I want you guys to do is go ahead and read the section I put up for mutations. Don't read the section that says, don't read this section because this we already talked about and I don't want to confuse you and give you any more stuff. So just read the section that starts at 8.7 mutations. You don't have to take notes on it, just briefly read it so that when we come back on Friday, I can explain the details. But basically on Friday, we're going to learn about mutations, and then we're going to do our discussion board on how mutations work. Is that clear, guys? So all you have to do is preview the section on 8.7 mutations, and please go back and rewatch today's video. All we talked about was step 1 of DNA replication. And then on Friday, we'll continue step 2 and 3, and then we'll do our discussion board. Sound like a plan?
Yeah.
Okay. All right, guys. So I'll go ahead and end class early. We're 6 minutes early. Like I said, I'll always record these now and put them on Schoology under updates so you can rewatch what it is you missed, okay? If you have any additional questions, or if you didn't get a chance to send me your photos, I'll be available all day today till 3. And again, tomorrow I'll be available from 3 to 3. If you want to talk to me, you can do Zoom. We can do just messaging, email, whatever works for you. Okay, but please, Oscar, April, and Thanos, send me your DNA models today. Okay?
Okay.
All right, guys.
Okay.
Have a nice day. I'll see you Friday. Bye, guys. I'm going to end class.
Bye.
Later, Mr.
Bye, guys.